Hello everyone, welcome back. So this is week 3 question answer session of the NPTEL course Fundamentals of Protein Chemistry, and the course coordinator is Professor Das Gupta, who is a professor at the Department of Chemistry in IIT Kharagpur. I am Beas Mukherjee, a research scholar from Dr. Shomude's lab of Structural Biology and Protein Engineering, and our department is Department of Bioscience and Biotechnology, IIT Kharagpur. Okay, so in this session we are going to discuss the question answers of week 3. So today's topic is protein folding. Okay, so I'll go through some theory portion and we will do the questions, corresponding questions, and this is how we have done all this while, so you are well familiar by now. Okay, so if you have any questions, you can ask me. You can type in the chat box or you can unmute yourself and ask me. Okay, if I'm not able to answer your questions, then I'll get back to you in the next class. Okay, so let's begin with today's class. So in this today's class, we are going to talk about protein folding, and we are also going to look into some diseases that are caused when these proteins in our body get mutated. So there are a lot of different diseases that are caused when proteins get mutated. Okay, so protein folding is one of the very important topics for your exam, be it NPTEL exam that you're going to appear for, or any competitive exams, for example, GATE, CSIR-NET, DBT, ICMR, anything. Okay, so protein folding is an important aspect there. Mutations in proteins, and there are also many other topics that will come up as we go through the today's session. Okay, so let's begin. Okay, right. So if there's any problem, please let me know. All right. Right. So in last class, we had talked about, in the first class, we had talked about amino acids. In the second week, we had talked about how these amino acids come together to make proteins and how these proteins fold into their native secondary and tertiary structures. Right. So now let's just go back there once again. So there are these very important interactions that go on in the 3D structure of a protein that makes the protein functional, right? The signature sequence of a protein is its amino acid sequence, which is making up the primary structure. Now, how these amino acid sequences, they are going to interact with one another, these individual residues, they are going to interact with one another. That is going to make the protein fold into its definite native structure. That is actually still a very surprise to scientists. Okay, so that is what we are going to explore today. So see, amongst the different interactions, there are hydrogen bonds. There is hydrophobic interaction. Hydrophobic interaction is between the, there can be van der Waals interactions, and hydrophobic interactions are mainly between residues that are mostly packed within the core of the protein. And then there can be disulfide linkages between cysteine residues. There can be ionic bonds and so on. So here is a list of different interactions, and you can go through this list. So it explains everything in details, okay, for your better understanding. For example, hydrophobic interaction. So these non-polar amino acids aggregate to avoid water, stabilizing the protein core. So this is one of the most important interactions in protein folding. So we'll read about that in details, okay, in the upcoming slides. So see, for example, leucine, isoleucine, valine, etc. cluster inside the protein. Then comes hydrogen bonds. Hydrogen bonds are very important to make the protein soluble. Okay, so when you want to solubilize a protein, let's say a protein is not very soluble and you're making it in, let's say, bacteria or something, and you want to solubilize the protein, then you introduce more of these residues that will undergo charge-charge interactions. Okay. So hydrogen bonds between the backbone atoms or side chain, important for secondary structure formation. So they are very important in alpha helices. Now van der Waals interaction, these are weak forces, closely packed atoms, and helps in fine tuning the structure. Okay. Now close packing of the side chains in the protein cores, that is done by van der Waals interactions. Now electrostatic interactions and salt bridges, attraction between oppositely charged side chains. So salt bridges, for example, there is a basic residue and an acidic residue. Let's say there is a lysine, which is a basic residue, and an acidic residue, for example, aspartic acid. Now these two are there in a helix and they are at a particular distance. We had studied about helices in our last class, if you remember, that these salt bridges, they are best when the first residue forms a disulfide bridge with the let's say fourth or the fifth residue. So I plus 3 and I plus 4 interactions. This is I and I plus 3 and I plus 4. These are best for salt bridge formation. Right. So that is how you can stabilize a helix. Now disulfide linkages. This you understand. There is a covalent bond between the cysteine residue stabilizing the protein structure. This is very important and very widely seen in a lot of proteins in our body. Okay. And it immediately improves the thermal stability of the protein. Okay, so the protein does not get destroyed even if it is heated at a very high temperature. Then comes pi-pi interaction. So there is a pi electron cloud in the aromatic amino acids. They are having this stacking interactions. Pi-pi stacking interactions of aromatic side chain contributes to stability. Stacking of the, let's say, phenylalanine residues or let's say tyrosine residues. So these are very interesting. Then there is stacking in DNA also. The DNA bases, you know, the way they stack, that is what gives DNA its stability. So DNA can be heated to a temperature of 98 degrees centigrade, right? It will just denature, but then the structure wouldn't break. It will not degenerate. So cation pi interaction. So this is also very interesting. So see, there can be interaction between a cation and an electronic pi electron cloud of an aromatic ring. So for example, interaction between lysine and tryptophan side chain. So all of these interactions are very important. Now, based on your understanding until now, you can try to answer these two questions. Okay, right. So you can type in the chat box. So the first question is, which of the following residue pairs does not form a salt bridge in protein? Okay, if you remember a salt bridge, it is between a positively charged and a negatively charged residue. Right? So which of these residue pairs will not form a salt bridge? Okay. So this is the first question and you can try answering the first question. And for the second question, which of the following options contain all the information necessary to fold a polypeptide chain into a three-dimensional structure? Right. So how does a protein understand? Initially, it is just a long chain. That is how after translation it is formed. Now, how does the protein understand exactly which way it needs to curl and which residues need to interact so that it will get its definite shape and three-dimensional structure which is going to make it functional. So how is the protein going to understand that? So that is your question. Okay, so you can try answering. These are simple questions. Based on whatever we have discussed until now, you should get an idea about these. Okay? Try solving. Let's look at the first question. So I'll give you a hint. See, glutamic acid and leucine. Now leucine is not a charged residue, right? So leucine is having all what? Aliphatic side chain, right? Aspartate and lysine. This is the example that I gave you guys, right? So this is, yeah, correct. Very good. So for the first question, the answer is leucine. Because for the rest, you see there is aspartate and there is arginine and there is glutamic acid and there is lysine. So all of these are having side chains which can get charged, which can become charged. So for example, lysine and arginine, they are having NH3 group in their, NH3 plus in their side chain. And aspartic acid and glutamic acid, the name itself tells you that the side chain is having acidic groups, right? So the answer would be A. For the second question, you try to attempt this. Second question is also simple. So what in a protein tells you how the protein is going to get folded? DNA sequence, histones, RNA sequence and amino acid sequence. C, yes, very good. That's correct. Yes, so the native conformation of the protein is determined by its amino acid sequences and the salt bridge arises from the anionic carboxylate of aspartic acid or glutamic acid and the cationic ammonium group from lysine and arginine. So leucine is having aliphatic side chain. That is why it is not going to be involved in your disulfide linkage. Sorry, salt bridge formation. Okay. So now there is one more question. What type of reaction occurs when a disulfide bond forms? So disulfide bond is formed by what kind of a reaction? So disulfide means that initially this cysteine residue is having SH and SH, that is thiol groups. Now this H is removed and the two the S actually form a bond. So what is happening there? C is oxidation. Yes, that's correct. Very good. Yes. See, disulfide bonds which are often formed between two free thiol groups are sulfur-sulfur bonds. The disulfide bond is in the oxidized state, whereas the free thiol is in the reduced state. Right? Okay, very good. Now we'll move on. So now we will look at what happens when proteins get mutated. Now that is something that is a real life problem. Right? There are so many different diseases in which are caused because of mutations in these proteins. Right? So one of the very common examples, and you guys have already read a lot about this. This is sickle cell anemia. Right, so what happens in sickle cell anemia? See, in normal hemoglobin, this is the structure of normal hemoglobin. It is disc shaped. But in sickle cell, it is like this thing with which you cut grass. That is sickle, right? This crescent moon kind of a shape. So what happens is there is a very small point mutation. Point mutation means there is a mutation in one nucleotide in DNA. So the DNA, see, this was supposed to be GAG, which is supposed to give you glutamic acid. GAG is in the codon, right? It is supposed to give you glutamic acid. Instead, it becomes GTG, and that codes for valine. So because of one single point mutation, the protein that is transcribed, which is, see, this HBSS. This HBSS protein is the sickle cell hemoglobin. And what is the difference there? The sickle cell hemoglobin is different from the normal hemoglobin, and the replacement of this glutamic acid with valine at position 6. Position 6 means in the protein there is the, right, okay, in the nucleic acid. In the nucleic acid there is this position, and then what happens is makes the beta chain more hydrophobic. Now hemoglobin is made of 2 alpha and 2 beta subunits, right? It is a quaternary structure. So 2 alpha subunits and 2 beta subunits. So alpha is normal, but then the beta subunits, it is labeled as beta S, beta sickle cell. So this becomes more hydrophobic. Okay, yes, typing something? No. Okay. So it becomes more hydrophobic, and under low oxygen condition, this hydrophobicity causes the hemoglobin molecule to come and stick together, forming long rigid chains that distort the shape of the red blood cells into sickle or crescent shape. So these sickle cells are less flexible. Now hemoglobin needs to be very, very flexible because it is supposed to pass through very narrow blood vessels, right? It is supposed to pass through that very narrow blood capillaries. But then the sickle shaped blood vessel cannot pass through it, leading to blockages that prevent blood and oxygen from reaching parts of the body. Now, being more fragile, they break apart very easily, leading to chronic hemolysis. Hemolysis, he means blood and lysis means breaking. So, red blood cells are breaking up, resulting in anemia, severe anemia, and severe pain and organ failure due to insufficient oxygen delivery are hallmarks of this disease. So one single mutation in the codon can give such drastic effects. Now, based on your understanding of this, try to answer this question. A point mutation in hemoglobin causes sickle cell anemia by changing a glutamic acid or glutamate residue into a valine residue. Which type of protein structure is affected by sickle cell anemia following this mutation? Right? So your options are the primary structure, secondary structure, and tertiary structure. So 2 will be correct in this case. So every option has C 1 and 2, only 3, 2 and 3, 1, 2 and 3. So which one do you think would be the right answer? So try answering. So one of the answers, yeah, okay. So you're saying that it is D. So see, why is that so? Because since it is changing, so glutamic acid to valine, so that means definitely primary structure would definitely change, right? What else is changing? That is basically the question. So yes, that's right. So the fourth option is the correct option, because changing a charged amino acid to a hydrophobic amino acid will disturb all types of interactions in the protein. Hence, it will affect the primary, secondary, and tertiary structure. So we saw that the tertiary structure was affected. We saw that in the figure, right, in the previous slide. Primary would of course get affected because you saw this particular mutation, so glutamic acid to valine. And another thing is it is becoming more hydrophobic. So even the, and so what is happening? Even the secondary structure is also getting affected. Okay, now we'll move on. So here for your, one second. Hmm, 19%. Okay. So for your knowledge, I have added this article and it's published in Nature Education. So you can go through these. So there are many diseases besides the sickle cell anemia where mutations in the proteins result in drastic changes, okay, in the body. So this is one of the sources from where you can read about this. It is of course going to be there in the textbooks, so you can of course refer to that, but this is one of the sources that you can look into. Okay, and here is a table just for your knowledge. So people will like all these different diseases such as sickle cell anemia, Duchenne muscular dystrophy, and all of these diseases that are there are questions from these. Okay, so I'm just giving you this as a gist. You can look into it in details, and when you start solving the questions, you will see that you'll come across these questions mentioning these diseases. Okay, so for that it's good if you just make small notes and know what mutation leads to what, and what kind of problem is seen in these diseases, right? What kind of mutation, what changes in the protein structure and phenotype and everything. So for example, see, there is missense mutation. Missense mutation, you know that the one that we saw in case of sickle cell anemia. So because of the change in the nucleic acid, a different amino acid is made. That is missense mutation. If the same amino acid would be made, because codons are degenerate, right? So 2, 3 codons can code for the same amino acid. So then it would be called nonsense mutation, right? So because of a missense mutation, there is sickle cell anemia in the beta globin hemoglobin, right? What is a nonsense mutation that I told you? So premature stop codon is coming. So there can be truncated protein. So let's say that, no, one second. Sorry, did I make a mistake in telling you what is nonsense mutation? You can just look that up. Okay, right. So in case of this nonsense mutation, a stop codon is introduced. Okay, so what happens is the full protein is not synthesized, but only a part of the protein is synthesized, and that is seen in Duchenne muscular dystrophy, where this truncated dystrophin is unable to stabilize the muscle cell membrane. So you can read about this in details. Then there can be frame shift mutation, and this is seen in case of this Tay-Sachs disease. So there is a difference in the frame, completely different amino acid is read because of this, and the effect is ganglioside accumulation due to this enzyme defect. So all of these different mutations are there. You can go through them and you can see what happens in case of which mutation. Okay. Right. So now we move on to something called the Levinthal's paradox. So what happens in Levinthal's paradox is, now people, when they started studying about protein folding, they realized that it is impossible for a protein to fold into its native structure within the small amount of time that it takes, right? Because let's say that, to give you an example, Levinthal made this paradox. He said that let's say there is a protein made up of 60 amino acids. Let's say that 10 different conformers or amino acid in the unfolded state. So conformers means, let's say this is an amino acid. It is having all these different bonds and the different side chains are there. So it can be present this way, 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 right? So different bonds can be turning in different direction. That is, different rotamers can be there for each bond. Because of that, different conformation, different 3D orientation of each amino acid is possible. These are the different conformers. Okay, so he is saying that, let's say that every amino acid can have these 10 different conformations. That is possible. So then there can be 10 to the power 60 non-native configurations of the protein, because there are 60 amino acids. Each amino acid can be having 10 different conformers, right? So then 10 to the power 60 non-native conformations can be sampled by the protein. Then assume that in 10 to the power minus 11 seconds per configuration. So for every configuration, even if it is taking 10 to the power minus 11 seconds, then 10 to the power 49 seconds would be used to go through to sample all of these different conformations and come to the native state conformation. And that time would be 10 to the power 42 years. So if every protein based on its amino acid sequence starts to sample every conformation possible, then the proteins in our world would never fold. But that does not happen. So then how does it happen? Okay. So Levinthal's paradox, the number of non-native configurations outnumber the native configurations by such a large amount that the proteins cannot fold on a reasonable time scale if the search in configuration space is unbiased. So that means he is saying that there is some kind of bias. So let's say the protein is initially this linear chain and it folds into this structure. But in between there can be so many different conformations. Now the protein is biased towards only certain conformations that it will sample before it forms this native state. That is what he is saying here, right? So how does the protein understand which is its native conformation and which are the non-native conformation? So that it does based on this energy funnel, the one that you can see in this figure. So see, what happens is initially the protein is in its unfolded state, that is shown as U in this figure. So this U is the unfolded state. Then what happens, the protein can traverse different paths and come to the native state. So the native state is the biologically active state. So native state is highly stable. So stability and energy are always inversely proportional. So if it is highly stable, it will have very low energy, right? So see, this is the free energy, this is how it goes, and this is the degree of nativeness. This is how it is going. It is exactly reverse of one another, and this is the conformational space. So initially the unfolded protein is at the top of the funnel, so it can go in any direction and it can form these different non-native conformational states, and it can also come to the native conformational state. So as it comes to the native conformational state, the degree of nativeness increases, the free energy decreases, and the stability increases, right? So free energy tunnel represents the decrease in the free energy and entropy in the unfolded state. So what is this entropy? Entropy means the degree of randomness in the system as the unfolded protein undergoes folding. So the number of the conformational species decreases and the semi-stable intermediates are formed. Ultimately, the ones that you can see in that image, right? They are forming this tiny what do you call this? The local minima. These are called the local minima, and the one in the center that you can see, this forms the global minima. Okay, so ultimately the highly stable functional native state is attained. Right, so we will look at this figure in more details now. Okay, you can have a look at this figure and we will read about this in more details. So see, initially what is there? There is this unfolded state. So the polypeptide chain has no stable secondary or tertiary structure. It is highly flexible and has a random coil conformation. It is just formed after translation, right? Now what happens? Nucleation. So So nucleation means there is a small segment of the protein that adopts a native conformation, which then acts as a nucleus with one or more secondary structure and element for the folding of the rest of the protein. So it begins at a small point and then it just keeps on increasing and the rest of the protein get folded. So that is one of the way of doing it. Then there is propagation. So after nucleation, the folding process continues as the rest of the polypeptide chain adopts its correct conformation. Then there is hydrophobic collapse. This is the most important one. This is the central reason your proteins get folded so easily, right? So hydrophobic collapse means the protein rapidly collapses into a compact state due to the hydrophobic effect. So in this state, the hydrophobic residues are buried within the protein away from the aqueous environment, which drives the folding process. Then there is collision collapse. So these are all different theories, right? Collision collapse. So partially folded proteins collapse into one or more compact form due to hydrophobic interaction and other forces such as van der Waals interaction, electrostatic interactions. Then there is the biologically active native state. The native state is the final and fully folded conformation of the protein where it is functional and stable. In this state, the protein has a specific three-dimensional structure that is critical for its biological activity. So if we go back to this figure, you will see that this is how the protein folds. So it can go through three ways. One is nucleation and then propagation. It can go through secondary structure formation and then collision collapse. Or it can go through hydrophobic collapse and specific structure formation. So these are the three different ways that people propose that the proteins get folded. Now, what happens is, see, initially the protein is, okay, so protein solution thermodynamics. Now in protein folding, something that is very, very important comes in is thermodynamics. So just now we read that this entropy of the unfolded state is very high because let's say this is the unfolded state. It can move anyway, so it is having very high degree of randomness, but then the folded protein, it is relatively having less randomness, right? So there is a definitely entropy reduction. Plus there is also an enthalpic component because bonds are being made, bonds are being formed and bonds are being broken. So there is constant interaction between the residues. These interactions are leading to this enthalpic component, right? So see, protein solution thermodynamics. So water molecules around the hydrophobic groups ordered to enhance mutual interaction. And hydration entropy is negative, hydration heat capacity is positive. So what happens is, let's say that there is a monomer and this monomer is forming a dimer. If you don't think in the form of a monomer, if you think this in the form of a normal unfolded protein to a folded protein, then also this is applicable. So what happens is, initially when it is an unfolded protein, the water molecules are arranged around it, or let's think in this term only. We'll think this in the form of a monomer and then it forms a dimer, right? So initially when this is a monomer, so see, release of the water molecule facilitates aggregation and folding. So the water molecules were there around it, and then what happens when it forms a dimer, there is displacement of these water molecules. So the degree of randomness in these water molecules get changed and the hydrophobic interactions make the overall entropy of the protein decrease. So there is a compensation between the water molecule and the protein that is resulting in the net. It is making it favorable based on the thermodynamics, right? So hydration entropy is negative and hydration heat capacity is positive. Now see what happens here. So when the exact reverse happens, so in the lab, it is very difficult to study how an unfolded protein makes a folded form. But then in the lab, what people do is they take a folded protein and then they unfold it. And this is rather relatively easier to study. So see, when the temperature is increased, this hydrophobic hydration is enhanced. So unfolding is studied, and when unfolding is studied, this favoration of net hydration disfavored by intrapeptide interaction. So solvent matrix expands upon heating, the population of the solvent around the solute decreases and the solute-solvent attraction is also weakened. Hydration entropy is negative. Hydration heat capacity is positive. So increasing temperature, what happens is hydrophilic hydration weakened and hydrophobic hydration weakened. So this is something very important and this you can read from the materials that I have provided. Right, so what happens is a thermodynamic aspect of protein folding. So this is very, very important. So what happens is that there is a redistribution of the water molecules. So therefore what happens, the entropy of the water molecules get changed and the entropy of the overall protein gets changed. This is actually making the entire process favorable. Okay, so the entropy of the water molecule is actually increasing because initially the water molecules were interacting with the surface of the protein, so they were rather more ordered. Then what happens when the protein gets folded, or here it is forming a dimer, these water molecules get dislocated, so their entropy increases, but the entropy of the protein is decreasing because it is becoming more ordered. So the net entropy of the system is actually decreasing 1 minute. That is why this process is getting favored. One second. Okay, so we'll move on. Right. So here we'll look into this process of protein folding in more details. So folding happens, you see that this is a very, very vast and rapid process. Now the thing is that for the proteins to do it alone would not be a very practical job, right? So that is why a set of proteins have been employed for this purpose. Now their function is to make sure that once the protein is synthesized from the ribosome, they're taking this nascent polypeptide which is just unfolded, and then what is happening, they are helping in its folding. So these proteins are called chaperones. Plus their also role is to make sure that you check, it's their role is to check whether the proteins are folded correctly or not. If these proteins are not folded correctly, they will send them back to the endoplasmic reticulum and make sure that the proteins are folded correctly. So this will be done again and again repeatedly until it is confirmed that the proteins are correctly folded. Only then these proteins will be allowed out and the proteins will travel to wherever they have to. Otherwise, what will happen is, so otherwise what will happen is these proteins will be labeled with these ubiquitins and it will be sent for proteasome mediated degradation, right? So protein folding is mediated by chaperones, which are heat shock proteins like HSP70 and chaperonins, for example GroES and GroEL in E. coli. So unfolded and misfolded proteins cannot pass the ER-Golgi quality check and are degraded into constituent amino acids by proteasome mediated degradation. So misfolded proteins can cause disease conditions such as cystic fibrosis if accumulated amyloid fibrils or insoluble aggregates, several neurodegenerative disorders such as Alzheimer's, Huntington's disease, etc. So these are the different kinds of diseases that can be caused because of aggregation of these misfolded proteins. And these misfolded proteins can be present in the form of amorphous aggregates, oligomers, amyloid fibrils, etc. Right. So how do these proteins fold into their native state? So see, it's given very nicely in a diagrammatic format. So GroEL and GroES, these are GroEL is basically like a chamber, and what happens is it uses ATP and the protein goes in, and this GroES closes it like a lid and then helps in its folding process. Now what happens is this HSP70 binds to the hydrophobic regions of the nascent misfolded protein and helps them fold correctly to prevent aggregation, and HSP90 assists in the maturation of the specific proteins involved in signaling and other cellular processes. Now, there can be certain kind of aggregates and certain kind of misfolded proteins that are seen in different diseases. For example, tau. Tau is seen in case of Alzheimer's disease. Huntington is seen in case of Huntington's disease, and alpha-synuclein seen in case of Parkinson's disease. So these you have to remember. Amyloid beta is seen in case of Alzheimer's disease. So these are something that get asked in questions. Okay, and this is something that you have to remember. These can come in the form of match the followings or any other different form. So you have to read about this properly and you have to remember this. Right? So now this quality check of the protein, this is very, very important. Right? So this is something that is very important because otherwise what happens is these misfolded proteins, they can cause different kinds of diseases such as cancer, neurodegeneration, abnormal inflammation, etc. So to ensure that these misfolded proteins, when can they be formed? So misfolded proteins can be formed when there is a huge stress going on in the system. A lot of proteins are being produced at the same time. There can be physical stress such as heat stress, oxidative stress, and translational stress. There can be mutation in the DNA, for example, exposure to UV light. There can be missense mutation, frame shift mutation, non-homologous end joining mutations. Then there can be protein overproduction, the one that I told you just now. So gene expression alterations. And there can be immunogenic insults. So for example, viral and bacterial infection. All of these different external and internal stress factors can result in these misfolded protein. So what happens is normally when a cell is having too much of misfolded protein, the body is equipped in this way that that particular cell will undergo autophagy. So the cell will just make sure that it is destroyed. Okay, and the cell, okay, so the proteins, all of this will go into this. So the cell can undergo apoptosis, or all the proteins will go into this proteasome mediated degradation or autophagy. So it will be eaten up, and then the tissue and everything else will be preserved that way. Right? So that is a kind of quality check devised by our body. Now see, here everything is given in details, whatever we discussed just now. So now protein denaturation and protein folding. So this is something very important. So what is the difference between protein denaturation and protein unfolding? So denaturation is a reversible process, but unfolding is generally irreversible. So what is denaturation? Denaturation means that, let's say this was the native structure. So we want this native structure to go back to the initial primary structure. So that is denaturation. But unfolding means that it is caused by agents that cannot be, like, it is a rather harsh thing, right? So what happens is transition from native to a more extended conformation, whereas denaturation is broad term for the loss of the native structure and function, often triggered by changes in temperature, pH and ionic strength. But this denaturation can be resulting from changes in temperature, pH, or presence of denaturing agents such as guanidine hydrochloride. So when we purify proteins in a laboratory, we also use these different agents such as urea and guanidine hydrochloride to completely denature the protein, so that all the interactions of this protein, all the native interactions, are lost. Okay, because let's say that your protein is binding to a lot of DNA. and you want to purify this protein and you want to perform DNA binding assays. And sometimes what happens is the proteins are very difficult to purify in their natural form, because let's say they are having this intrinsically disordered regions which have this propensity to form random coil, and these can get misfolded, they can go into inclusion bodies, all of these different problems happen. So for that, the proteins, they are actually completely denatured, so only the primary sequence is retained, and that is how they are purified. And when you're purifying, during the process of purification, these are once again refolded into the native form. Okay, so now unfolding, see what happens. It is reversible and can occur using mild changes in condition. Thermal unfolding of proteins at moderate temperature, but heat induced denaturation means that you're heating it to, let's say, 95 degrees centigrade or something, and the protein is not a thermostable protein, so then completely it is denatured. So that is how it happens, right? So now see, uh, these are important graphs. So denaturation, a loss of the three-dimensional structure sufficient to cause loss of function, not necessarily means complete unfolding or random conformations. An abrupt unfolding over a narrow temperature range, cooperative unfolding process can also be seen. So denaturing agents can be heat, that is weak hydrogen bonds are broken, because of that pH alteration, so alteration of the protein net charge. If the net charge is altered, definitely the folding will be affected. Alteration of the electrostatic repulsion and disruption of hydrogen bonds. Now organic solvents such as alcohol, acetone, urea, guanidine chloride, detergent, etc. These can also be used, and disruption of the hydrophobic interactions. So these mainly target the hydrophobic interactions in the protein. Okay. Right. Now we move on to protein crystallography. So I have included all of these topics because very soon we have questions coming up, and if you have some understanding of these topics that we have discussed until now, just from the next slide the questions will begin and you will be able to answer those questions. Okay. Right. So we'll start with protein crystallography. So how are crystals made? The protein crystallography is very difficult process, and you can see in this figure, these are the protein crystals. This is how protein crystals look like, and then X-ray is passed through these crystals, and the proteins in the crystals, they actually diffract these X-ray, and then the diffraction pattern is studied, and based on that electron density map is created, and based on the good resolution of this electron density map, what amino acids are present in which position, that can be found out. Okay. Now crystals depends upon protein concentration, protein quality, stability and purity. Crystallant, that is salt, precipitant, pH and buffer, these are required for crystallization. Their specific concentration is very important, and the different physical variables such as the size of the droplet, ratio, temperature, setup, everything else. So you can read about this in details. Here this is just to give you a brief idea of what happens in X-ray diffraction and crystallography. Okay. Not all proteins are crystallizable. Now crystallography is very important and very useful for proteins that are very well folded. Now I was talking about these intrinsically disordered regions in proteins, which are also very important because these help in saving up a lot of energy. So they are largely unfolded, and they are very important sites for protein-protein interaction and protein-DNA interaction. Okay, so now it is very difficult to crystallize these proteins because these are so random and they are so flexible that you will see in the protein data bank that all the crystal structures of the proteins, they will largely have these alpha helices and beta sheets. Just that portion, just domains. But then the N and C terminal regions will be missing. That is because these are very flexible, and very flexible proteins are very difficult to crystallize. Okay, so protein crystals are exposed to X-ray. Now X-ray can be in-home source or it can be a synchrotron. Okay, now which diffract as they pass through the crystal. The resulting diffraction pattern is recorded and analyzed to get the electron density map. The structure determination and refinement process is to get the 3D model. So this is done. It's an iterative process and it takes a lot of time to do this. So crystallographers are really patient people. Okay. Now we'll move on to the week 3 assignment questions. Right, so try to solve this question. So this is a graph that tells you from the above mentioned curve the melting temperature of the protein will be. So how do you calculate the melting temperature? This is the temperature axis and this is the fraction of the unfolded protein. Okay, so the melting temperature is actually the midpoint of this log phase. So that is how you get the melting temperature. It is the temperature at which 50% of the molecules have melted. 50% is melted. That is your Tm or melting temperature. Now let's say you have stabilized a protein. Then what we will expect is that the curve will shift. So there will be this rightward shift and the melting temperature will be rather higher for this stable mutant. Okay, so from here, what do you get as the melting temperature? So it's going from 0 to 1 C. Okay, right. Yeah, let's check. Yes, that's correct. C is the melting temperature. So see, the values are going from 0 to 1. And so if you go to 5, that is 0.5, which is 50%. And then if you just come to this point, then you see that if you draw the line and make it meet at the x-axis, then you will see that it meets at 45 degrees. So at 45 degrees, half of the protein is unfolded. Half the population of the protein is unfolded, right? So its melting temperature is 45. Now we go to the next question. What is obtained after the X-ray diffraction of proteins? Molecular density map, nuclear density map, electron density map, and atom density map. C, yeah, very good. That's correct. Electron density maps. You are the only student who's here. The rest of them have left. Okay, no problem. So which of the following amino acid sequences is most likely to be the part of the core of the protein? So there are these amino acid residues, and which of these amino acid residues do you think would be most commonly present in the core of the protein? So for the core of the protein, you will have to look at the residues that are more hydrophobic, right? So for that, you have to remember the single letter codes of these amino acids. For example, K is lysine. Now lysine is a charged residue. It is more, yeah, correct. Yeah, that's right. So B would be the right answer. That is correct. Yes. So phenylalanine, histidine, methionine, cysteine and valine have non-polar and hydrophobic side chain. Hence, phenylalanine, histidine, methionine, cysteine and valine tend to cluster in the interior of the protein. Right? Now we go to the next one. Now, molten globule state of a protein. Now, molten globule is something that is even more stable compared to the native state. Okay, it is one of the states in the folding process. Which one of the following option is incorrect about the molten globule state? So, which is incorrect? It is a denatured state. There are hydrophobic interactions among non-polar residues. It is less compact and it is a collapsed state. So which of these do you think is incorrect? These kind of questions, the more you practice, more well versed you will become with these kind of questions. It will be easier for you to solve. let's look at the answer. So the answer is denatured state, because molten globule is partially denatured state where the interior side chains are improperly packed and less compact than the native state. Okay. Right. Now, so protein folding is a spontaneous process. Spontaneous means that it is energetically favorable. So delta G is positive, delta S and delta H are both positive, delta S is negative, delta H is positive, and delta S of water is positive and delta H is negative. Right. So this delta G is your Gibbs free energy, delta S is change in enthalpy, change in entropy, delta H is change in enthalpy. Okay, so the formula is delta G is equal to delta H minus T delta S. So if this T delta S factor becomes big, only then the entire thing would become more negative, and so the delta G will be negative. So if delta G is negative, then it is a spontaneous process. So the free energy is negative, that means it's a spontaneous process, it is energetically favorable, right? So what happens here? D, that is correct actually. That is correct. Yes. So in protein folding, I told you that entropy of the water is positive. That means that change in entropy of water is positive. That means water was initially more ordered and from there it is getting displaced. So entropy of the water is more positive, but entropy of the protein is more negative, and enthalpy is negative because it is all these bonds and new bonds are getting formed, and that is why a lot of energy is released. It is exothermic and it is largely negative. Because it is negative and delta S of the water is positive, all of this is actually making the delta G more negative. Right. So in the protein folding, hydrophobic collapse of a polypeptide chain leads to an increased entropy of water. Change in enthalpy is large and negative. This is the overall change in enthalpy. It is large and negative. Why? Because hydrogen bonds, electrostatic interactions, overall change in enthalpy, right? Interactions, van der Waals interactions contribute to enthalpy, and protein folding is driven by enthalpy. Okay. Now, which class of proteins is a heat shock protein part of? So, heat shock protein comes under catalytic protein, chaperone-like protein, immunoglobulins, ribonucleolytic proteins. B, chaperone-like protein. Yeah, correct. In the hydrophobic core of the protein, which of the following forces is responsible for the hydrophobic collapse? Now, this Keesum force, Debye force, and these, you will have to remember this dipole-dipole forces, then dipole-induced dipole. So, these are the different names given to these. Okay, and you will have to remember exactly which is which. So that is how you will be able to do it. So for hydrophobic core of the protein, which one do you think is responsible for the hydrophobic collapse? D, van der Waals force is correct. Yeah, that's right. So see what is happening. So van der Waals forces exist for all particles atom sized and up. So permanent dipole-dipole interaction is called Keesum interaction. Permanent dipole-induced dipole interaction is called Debye interaction. And induced dipole-induced dipole interaction is called London or dispersion interaction. This is always present. And these are all long-range forces. Right? So non-polar surface of amino acids build the hydrophobic core due to van der Waals interaction between hydrophobic and non-polar side chain. So there is hydrophobic interactions. Okay, these are not there. Now, so this is explained in more details. So hydrogen bonds, these are actually the bond length is given. Sometimes questions get asked based on the bond length also. Okay, so van der Waals interactions. So see, these are the bond length is this, and disulfide linkages are around 2 angstrom. So these are short range interaction. Salt bridges are also very short, they're around 1.5 or something like that. Okay, 1.8 angstrom or so. Okay, rest, these are all long range forces and they act. Now, the thing is, you have studied about protein folding. So right now, a lot of these different softwares and AI driven models and DeepMind and then all these deep learning models, these are all getting formed, and these are very effective in predicting the protein structure. So initially there was this homology modeling, there was fold recognition and threading and ab initio modeling. So these were based on some data set, right? So let's say that you are telling your computer that if this is the sequence of a protein, it is supposed to form a alpha helix. If this is the sequence of the protein, it is supposed to form a beta sheet. So initially, all of these training models were taken from protein data bank. Now, protein data bank structures, largely they were crystal structures. So then what used to happen is, all of the models would predict the folded region very well, because crystal structures means only the folded regions are there. The NNC terminal, these are not getting crystallized, right? So you don't have information about them. So all of these were giving very good data for the folded regions. But now AlphaFold 3 has come up, and this can even predict the interaction between a protein DNA, protein interactions, protein small molecule interaction, and then like it's very useful for any kind of designing any kind of mutant proteins, and also knowing the structure of any sequence of amino acids. So you can try this for yourself. You can just type AlphaFold server, be it DNA sequence, be it RNA sequence, be it protein sequence. You just type in a random sequence of a protein and then you see how it actually folds it. So it is even very appropriate for the NNC terminal regions, because now a lot of NMR structures have also been solved where these NNC termini have been solved. Then there are also other predictive models that have been able to generate these NNC termini. So now the data that we are feeding, that they have fed into this AlphaFold server, that is really vast and covers a wide range of proteins. So now the predictions that we get, these are very appropriate. Okay, so see, sequence structure function. So initially people thought that this is the sequence of a protein and this is the sequence of another protein, then they are supposed to have the same structure. And if they have the same structure, they're supposed to have the same function. But that is often not the case. Even if two proteins are having the same structure, they can have different sequence. If they're performing the same function, they can have different initial starting sequence, right? So that is totally possible. Now see, folding impossible, but for the smallest structures, and function prediction from structure is very difficult. Okay, right. So now we will move on. So what is exactly homology modeling? So homology modeling means that let's say you have a protein and you know that this protein has this particular structure, it has this particular sequence. And then you find another protein and you see that let's say that this particular protein, it is having similar sequence, then you're expecting that it could probably have the same structure and it would also probably have the same function. Correct. So that is homology modeling. That means you have already have a initial structure, a solved structure of a protein, and since the you're expecting that this one is also going to have the same structure. So if we go back to our previous slide, so used for proteins that have homologous protein structures deposited in the protein data bank. So you can try to solve this question now. The homology modeling algorithm is applicable for proteins belonging to the same family, proteins belonging to different families. Okay, C, proteins with similar amino acid sequence, proteins having the same secondary structure with different linkers. Okay. C, protein modeling is applicable by assuming that the amino acid sequences have a very similar tertiary structure. Proteins belonging to the same family have same secondary structures. The linkers may vary. So that is 1, 3 and 4 are correct. Okay, so proteins belonging to the same family, they are homologous and they are having a similar amino acid sequence, and therefore they will have similar secondary structures, right? So yeah, that is homology modeling. So that is all for this class, and these are the different references. You can refer to these different resources. And thank you. If you don't have any more questions, we can end the session. Thank you for your time. Thank you so much. So I'm ending the session then. So we'll meet again next week. Hopefully something useful you will find in the next session.